You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Lynn Caporale. She's the author of Darwin in the Genome, Molecular Strategies in Biological Evolution. She's a biochemist who's focused her attention on evidence that natural selection can act on mechanisms that generate genome variation. And this action can create things such as beaks and wings and other adaptations from creatures. So looking forward to speaking to her. So Lynn, thanks for coming. How you doing?
Good to be here.
Yeah. So if we could start out, what got you interested in evolutionary processes so long ago, and what's your journey been like as you've been studying it?
Well, I actually came in from a different direction. I was a chemistry major. I then became interested in biochemistry and was actually trying to design inhibitors of an enzyme involved in inflammation in the complement system. There were related enzymes involved in blood coagulation, and I didn't want to block them while blocking inflammation. And so I began to think about the relationship between or among proteins in a protein family and how they might have evolved, how specificity kind of becomes overlaid upon homology in a protein family. And and at the same time, I was reading about Shannon and codes and thinking about the genetic code. Actually, in somebody's qualifying exam, my mind wandered a bit and realized that because the genetic code is called redundant, in other words, or degenerate, there's different ways of specifying the same amino acid. You actually can send additional information through the same DNA sequence. And one type of information I hypothesized was information that would vary the mutation rate. So if you were evolving a new member of an active gene family, you might explore the active site that focuses the action of the protein rather than damage the structure of the protein, so that mutation would be modulated through evolution.
Well, okay, so a bunch of questions. I don't know if I'm expressing this properly, but if you have a given sequence of DNA, can that sequence be read at different starting and stopping points to create different proteins? Let's say it's 1,000 base pairs long. Can you read it from 1 to 500 and get one protein, and then 501 to 1,000 get another, or
Yes, well, that happens even in bacterial phages that have very compressed genomes. For example, the first genome sequence was of a phage, bacterial virus, a tiny genome, and they did find overlapping genes. But beyond the what you point out, the overlapping protein coding information, you can have additional information. So a protein coding sequence might specify a string of amino acids, but you could have additional information that might affect how rapidly that string of amino acids would vary in one region of it compared to the other region of the same protein, and other kinds of information. It could all be overlaid, and you might think you'd see a protein sequence. We know how to translate from DNA to through using the table of codons or the genetic code into a protein sequence. So we might think, oh yeah, we know what that sequence of DNA, quote, means, but there may be other information in there that we don't even look for because we think we know everything, right? But we don't. So as
Yeah,
we
because this is that's amazing. It makes you wonder how complex is our genetic code. I mean, it makes me think. I've heard like there's no such thing as a gene, or it's hard to define it, and maybe it's just that one reason alone that the same stretch of base pairs can code for different things in different ways tells you, well, there is no, this stretch of 500 base pairs, you can't call it a gene because it's not really read that way. It can be read many different ways or interpreted.
Yeah, and there are kinds of information in the genome that we may not be aware of. We're learning more over time, but there are just different levels of information running through the same DNA sequence.
Well, how do we know that there's different levels, and what do you mean? What kind of information? What other info could there be?
Well, one type of example would be that when you copy DNA to RNA in someone like us or an animal, there are these introns and exons. So the RNA says DNA is copied to RNA, and then the RNA is translated into protein. But the DNA copied to RNA, the original RNA is longer. There are spaces in the RNA that's spliced out before the so-called messenger RNA goes to the protein factory or ribosome to make the protein. So the parts that are spliced out, there is information at the edges of the protein coding sequence and even inside the protein coding sequence that helps direct the splicing system to make the cuts in the right place. So that would be another kind of information overlaid on the protein that might constrain. So there may be several ways to specify a particular amino acid in the table of codons, but which one of those triplets you use to specify the amino acid might be constrained by the need to send additional information, such as where the splice site is or other kinds of information.
So within DNA itself, there could be hidden levels of instruction to do things besides just transcribing that particular segment of DNA and making X number of proteins with it.
Right.
That's pretty amazing.
Yes, and the information can be. I mean, the other interesting thing about this other information is it can be represented in ways we don't know. So, for example, we think of DNA as A, T, G, and C. They're letters, but really they're not letters. They're physical chemical entities. And the proteins that interact with DNA or the RNA view it as a physical chemical entity. Obviously, they're not reading letters. So the information can be in the structure of DNA, its physical chemical properties, the relationship between neighboring sequences, rather than just in the letters that we, quote, read ourselves.
And, it just made me think of a protein. Proteins fold in certain ways, but can proteins fold in different ways and still have a stable configuration? Or I guess they could have different properties depending on how they fold, if they fold differently, right?
Well, certainly. I mean, proteins also a particular protein can have multiple conformations that can be manipulated by the small molecules in its neighborhood. So that's one way a small molecule might be able to regulate the behavior of a protein. So that molecule protein could be sensitive to the level of a nutrient might change the activity of a protein or some metabolite. So there's a tremendous amount of regulation of the behavior of proteins and DNA within the life of an individual. And what my work was focused on is how the genome changes over time, rather than just focus on the individual. Look at it over time, and then you begin to think about evolution.
Well, that's another question. How does a genome change over time? Is it more like epigenetics and expression of genes is what changes, or is it the underlying sequence of base pairs in us, for instance, that changes over time as well?
Well, ultimately, over time, of course, the base pairs change. You're right, epigenetic regulation is important too, but that's how you get from the original single cell organisms and then the initial multicellular organisms to people, right? That there are base pair changes in DNA. So then the question is, how does that happen? And a lot of people, I think one big misconception that people have, and there are a few I can discuss, but one is when they say random mutation, the assumption, the way we use random in our everyday life, is that any mutation is equally probable, but that's really not the case. And even Darwin himself, he really never said random mutation. In fact, I have a quote from him. He said, "I've sometimes spoken as if the variations have been due to chance. This, of course, is wholly incorrect, but it serves to acknowledge plainly our ignorance of the cause of each particular variation." The concept of random was introduced by the early statisticians, such as Fisher and others, who developed evolutionary theory long after Darwin. And but it's really not accurate, because if you think of the components of random, the first thing you would think, the casual use of the word random, is that anywhere in the DNA you could have an equal probability of any kind of genetic change. And that's not true. As a biochemist, that never seemed like a reasonable thing, because DNA is varied. A polymerase, the enzyme that copies DNA, can make a mistake, or the enzyme that repairs DNA might miss an error. And how likely that is really varies depending on the physical chemical properties along the DNA sequence, which any DNA sequence has varying physical chemical properties along it, just because it's made up of the A, T, G, G. And, GGGG is going to have different physical properties from AT. So this is a variation. And evolutionary theory says that selection acts on variations. So what I proposed is selection would act on the variations of probability along the genome, just as it acts on variations in beaks and wings. And so eventually there would be kind of a feedback control model of evolution, where the variation and selection are in a kind of a loop, and variation is not outside of that loop. It's that the genomes that generate variants that are more likely to survive are the genomes that will be more effective at adapting, and therefore would tend to survive. So, in other words, yeah, go ahead.
I mean, there's survival, but then there's, changes. How do creatures evolve from other creatures? I mean, so survival is just one thing, but variation seems to be a completely different thing.
Well, adaptation is so if you think of variation along lineages, we all think of contemporary populations. So in other words, you think of fitness in terms of, some people, when they think of fitness in Darwinian terms, they think of one fit individual who's reached the peak of fitness and is the strongest and toughest and so on. But the environment can change dramatically. And so that organism or individual who seems very strong at the moment may, in fact, if the environment changes, not be the strongest. And so what you want is a population of individuals that are diverse. So actually, diversity is a form of fitness. And then when you have this very diverse population of individuals, if some of them are always in one environment and some are always in another environment, they may diverge further apart, and you can begin to, call them. Eventually, they may become different species. But the main point I want to make here is that diversity is a form of fitness, something that gets missed a lot,
Well, when you say diversity, what kind of diversity within a population would be a signal of fitness? What's an example?
Okay, well, let me give a simple example from bacteria. So bacteria have coat proteins, proteins on their surface in addition to all the things they're doing. Let's talk about pathogens. So a bacteria can infect us, and our immune system will see the surface of the bacterium and mount an immune response against it. Now, suppose, and bacteria can divide and have descendants every 30 minutes, say. So suppose that the coat of the bacterium is very likely to mutate compared to, say, its internal proteins like DNA, the enzyme that copies DNA or something. So if you have a coat, now how would that happen? Well, if say you have a sequence of DNA that's CAG, 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 that's encoding the coat protein, or CAG, TCAG, a repetitive sequence, or CCCCC, then the DNA polymerase can sometimes slip and get out of line. It can miss a repeat, and then the protein will change, or it may be knocked out and a different protein appears on the surface because the bacteria has different, if we're knocking out an enzyme that's involved in constructing a surface feature of the bacteria. So the main thing I'm saying is if you have any one bacterium, its descendants will be very diverse on the coat. And that kind of diversity will protect it because the immune system will figure out how to attack one coat, and then the bacterium will make another coat. And that's how the Lyme spirochete actually is very effective. It keeps sticking patches into its DNA to vary its coat, so the immune system can't keep up with it. So that would be a form of survival. And it's also interesting that bacteria have what I would call reversible mutations. So there's one meningococcus, for example, there's a bacteria that needs one version of its coat to avoid the complement system, which is one of our antibacterial systems in our blood. But it needs a different version of its coat to stick to our tissue. So what's the bacterium going to do? It can either stick to our tissue or avoid the complement system. So what it does is it generates multiple descendants, some of which can avoid complement and some of which can stick to our tissue. So any individual bacterium will have descendants that can do both. So it can survive and its population of descendants can survive and stick to our tissue. So
why call this activity mutation? Why not call it adaptation? Because it's purposeful. It's directed.
That's very interesting. So it is mechanistically a mutation because the DNA varies. But the question is, but you're absolutely right. It's adaptive. This has been captured by the genome to generate variation in certain spots, and therefore genomes that are efficient at doing this are better at adapting and surviving.
And I guess another question is, who's calling the shots? Who's orchestrating this adaptation, or what is orchestrating the adaptation? Let's say in a bacteria, is there any? Have we been able to determine what is the driver of this?
Yes. So what's happening is you have a population. Say you start with a bacterium that doesn't vary its code. It has a high rate of variation somewhere else, perhaps, but not in its code. And you have another lineage of bacteria that generate these variations in the code. And then you infect an individual, and the individual's immune system will mop up the bacterium that didn't change its code. So that's why we call it selection. There's selection and variation, and the groups that are more the variants that are better at surviving are favored, and they survive, and then that lineage that generates the focus variation in the code is the one that survives. So that's kind of that's this feedback control model of evolution. So you generate variants, and they can be distributed just because of the biochemical variation, and then it becomes selected for and adaptive, and so the ability to evolve itself evolves.
I guess an odd question is, does there have to be? Okay, so in the bacteria, for instance, where is the bacteria's mind or brain? What directs this adaptationary drive? Or is it just an emergent property of the biochemistry, do you think?
Well, it starts with the biochemistry, yeah. the bacteria's mind. I mean, bacteria know things. We could say they know to swim. They swim towards certain things. They swim away from certain things. So within their lifetime, they have those kinds of behaviors that have evolved, because if you swim away from a nutrient or swim towards something that's toxic, well, those bacteria have died out, but then over evolutionary time, as you say, it emerges from the biochemistry. You have some intrinsic variation just due to simple biochemistry in the probability of different mutations along a genome. And when those mutations are aligned with the kind of challenges that the genome faces over and over, because the environment has structure, right? For example, a bacterium pathogen will continually face the challenge of our immune system. So when the environment has structure, the organism can evolve compensatory structure through this variation, selection, feedback loop over evolutionary time.
Yeah, it's just odd to me that there's a big school of thought that evolution's all random and purposeless, but yet everything else that life does is very purposeful. So why do you think people the rules are different for evolution, but for everything else there's a purpose? For this, no.
Right. I think there's a couple of things that are misunderstood. The use of the word random is definitely inappropriate given the common use of the word random. If you use a kind of a technical mathematical definition of random, which is probabilistic, but it can be uneven, that's different. But in our common everyday discourse, we use random to mean anything is equally probable, and that is certainly not appropriate for looking at genome variation. And it's just
the language is poor. It makes no sense why it would,
Yeah, and I think so. I think there are several things that are. One is the misuse of the word random, because over evolutionary time frames, lineages of organisms have gotten better at adapting. And secondly is the use of the word fitness, which I talked about a little bit earlier, that diversity is a form of fitness. And people in common everyday discourse think of fitness, you think of this one really strong person who's healthy, but in fact, fitness depends on diversity. You can imagine if a new virus swept through the community, if everybody was exactly the same and the virus was exactly matched to the people who were there, everyone could be wiped out. And we had that example in the early days of the HIV epidemic, before we had any protease inhibitors or other compounds to treat it, that the people who had this one rare mutation in a protein called CCR5, the delta 32 mutation, they just survived. Everyone was wondering, why are these individuals who clearly were infected or exposed to the virus, why didn't they get sick? And that's because there was a variant in the population and it protected them. So in general, the concept of diversity is important. I mean, I can give an even simpler example. In the old days, when we were all living in the forest, and you'd want some people who were early risers and some who stayed up late. So we'd always be protected in our population in the middle of the night. So, if something came to attack us, somebody would be awake, right? If everybody went to sleep at 10 p.m. and woke up at 6, we might miss something that came at 2 in the morning. So diversity in a population is a form of fitness. And another issue that gets mistaken is cooperation is another form of fitness. Again, we all think of ourselves as strong and competent and the greatest or whatever. Many people do. But the fact is, cooperation is important. For example, let's go to bacteria. Antibiotic resistance spreads from a bacteria that has an antibiotic resistance gene. It shares it with another bacterium. So that other bacteria doesn't have to evolve antibiotic resistance from scratch. And there are these plasmids or transposons traveling among bacteria that have resistance to almost every known antibiotic on them. So the bacteria cooperate and share this information. And of course, the human genome, if you go back a few generations or go forward a few generations, anyone's genome is really shared with, it comes from a lineage of thousands of people. It's created in the egg and sperm combination that created each individual, but then it goes on to those who have children and grandchildren. It gets split up again. So again, the human population is sharing pieces of the genome. So cooperation is really essential in evolution and in the fitness and carrying on of DNA over time. Cooperation, diversity, and then this feedback control rather than randomness are three important concepts.
What about is there any evidence of a new species or dramatic evolutionary changes that people have observed that are occurring right now, or is it just seems like, oh, it happened long ago and no one's seen it now, and why is that? And can you elucidate that a bit?
Well, I think speciation is a slightly more complicated issue than the evolution of a population. But I think the example I gave of HIV, if when it would spread through a community in the absence of any medical treatment, everyone who was not intrinsically, in other words, preemptively resistant to it, would have died, and many did, tragically. And so that would change within a few years, the genome of the shared genome of the community. And you look at why is there such a high incidence of sickle cell trait in populations in areas where malaria is endemic, and that's because if you get one sickle cell gene and one normal, what we would call normal here, hemoglobin gene from your parents, then you're resistant to malaria, but you don't get sickle cell disease. So if you don't have the sickle cell trait and you have two so-called quote normal for us who are living in a in a temperate zone without malaria endemic here, we go to we're susceptible to malaria, whereas a person who has one sickle cell gene and one normal gene would be stronger under those circumstances than we are. They would be resistant to malaria. So you see how even within the human population, there are going to be regional differences depending upon the challenges that communities have been exposed to over time.
But at what point would something more dramatic happen? Is it a pile up of changes over time until there's this tipping point where a new species emerges? Or, like, I'll give you an example. I went to a whale museum in Iceland, and they're saying, oh, the whales have vestigial legs and feet, and what they think is that, they were land creatures that walked back into the sea and became whales.
Right.
How would such a thing like that happen? Such a dramatic change?
Over extensive periods of time. So, I mean, if you go back to the hypothesis that certain regions of the genome may tend to vary more, I would, so for bacteria, you would say, oh, the coat proteins definitely are highly variable for interacting with the environment. For us now, I'm speaking about vertebrates in general, so that would include our friends, the whales, and the animals whose descendants were whales. Perhaps bone structure tends to vary a little bit more, so that we can explore adaptations of different forms of limb structure that would be useful in different environments. So it wasn't like the animal walked into the sea and its kids were whales. It happened over time, and eventually the animals separated from the ones who were on land and became better and better adapted to the environment there. And speciation is a more complicated discussion in terms of is it isolation? In other words, once you can't you can't reproduce, with that other organism than the species, then you're considered separate species, which is a kind of traditional definition of speciation. But I think rather than get into what technically is defined as speciation, I think you can just think in terms of generating variation that's appropriate to the environment, and then generating more variation that's appropriate to the environment, and that certain and then becoming so comfortable in that environment that you don't go back, and you couldn't go back. You've lost adaptation. For example, we we require vitamin C as the so-called limeys. As the British sailors discovered that you would get scurvy. So, but our ancestors a long, long time ago could make vitamin C, but we had it in our environment, and so it wasn't an essential trait, and apparently somewhere along the line we'd lost the ability to make it. So you
but
can
if people
see
tried to why haven't people tried to take fruit flies and breed them and pressure them selectively so they turn into something else? Maybe you can get 1,000 generations of fruit flies, and you can get them pretty far along into a different direction or a new direction so they turn into something else. I mean, is there any evidence that we've seen speciation in our time?
Well, I think what you're asking is, so if you define selection, so you pick what you want, what selective trait you're selecting for when you say put them in a different environment. So, for example, they've put bacteria in environments where they've stressed them for nutrients, and they found that certain genes will amplify. In other words, if they need more of a certain nutrient, they will amplify certain genes that will help them to survive under that specific nutrient stress. So the whole population, in the whole population, you will find the survivors have amplified a specific gene that helps them survive in that stressful situation. But that wouldn't be speciation in that case, because if you took the stress away, then they'd stop carrying all these extra copies of the gene around. They tend to lose it in that specific example. So when you want to put but you can, by selecting a population, you can make it adapt to members of the population will be favored who are able to adapt to the new circumstance you put them in.
But and I know it's a complicated question, but what's your guess? How did if we started in the sea, how did creatures leave the sea and never go back and be able to breathe on land, breathe in the air, or how did things fly? How did that happen? No matter how much you willed it, how do you adapt so that you can fly versus not fly? Where did birds come from?
Dinosaurs, actually. They came from dinosaurs. So that's an active area of research. Now, I'm a biochemist, okay, so that's not my area of expertise. But from what I've read, feathers may have initially been for heat, like your hair stands up on edge, maybe for heat protection or other kinds of not immediately to fly, but they were useful eventually to birds. But birds did come from dinosaurs, and people now talk about non-avian dinosaurs being the original time. But they've found the lineages, and I recommend to anyone who's interested in that specific question, a place like the American Museum of Natural History you can walk through there, and they'll take you through the current thinking about how birds evolved, to answer your question. But as a biochemist, and with this kind of more molecular-oriented or genomics-oriented approach to thinking about evolution, my thought, again, going back to that issue that vertebrates may tend to explore limb structure more, have more variation in terms of how long their limbs are, how thick they are, and so they would explore the adaptive potential of variations in limb structure. That would be one of the areas that vertebrates might explore.
Well, even any change, any adaptation, does it happen on the level of on an epigenetic level, where methylation is changed and the histone level, or does it happen on the DNA level where literally the sequence will be changed, or is it both? Where do these
Well,
things
there
tend to
are, I mean,
happen?
yes, both things can affect, it can affect the phenotype, what the organism is like, both epigenetic changes that are, and for example, even though changes to the sequence of the DNA traditionally has been the main focus of heredity, we do know that, for example, the famous example of the Dutch hunger, that that descendants of those who experienced famine during World War II did have effects on their metabolism, and that were passed down among generations. The fact that the parents had experienced a great famine, but the majority of changes over evolutionary timescales are definitely at the DNA level. So you can have variation both in regulation of genes, various epigenetic changes, heat shock proteins that may protect different variant structures, but over evolutionary timescales, definitely the dramatic changes are changes in the DNA.
So I guess in order for a change to change a species, it would have to occur in multiple instances in a given species almost contemporaneously, so that they could mate with each other and select themselves in some manner. It doesn't seem likely that there's a random change in one of the members and that created a whole new species because it was able to breed so successfully as to change the entire species. So it seems like, I don't know, it's just my theory, but I don't know.
Right, and so going back to the fruit flies that you mentioned, they have seen things where a piece of the DNA inverts, and then that messes up how the chromosomes interact during meiosis, so that only organisms that have the similar inversion can breed with each other. Even though if we looked at them, we'd say, well, they're all fruit flies, right? But so there are. But again, my focus is on the underlying biochemical mechanisms that generate diversity in the genome, and not on the issues that you've brought up. For example, biological speciation, which biologists, evolutionary biologists, really focus on. You've also brought up some of the issues of anthropology. So I don't claim to be an expert in those areas.
Well, how about, all right, so, I'm sorry to take you out of your expertise.
No, it's all interesting. I mean, it's all interesting. Yes.
Well, even within, all right, so biochemistry, that's your big thing. Have you or anyone documented at a biochemical level a creature that has a stress applied to it, and literally the steps and what changed in the biochemistry and what happened first and next, and the whole cascade that led to a change in the arrangement of base pairs of DNA or an epigenetic change? Have we even elucidated that mechanism biochemically in full?
Okay, so you've brought up another interesting point, which is the effect of stress. So people had assumed, again, taking into the word random, that genetic variation occurs kind of evenly over time, and there's even this concept of a molecular clock. But in fact, starting with Barbara McClintock and her study on mace, she observed that certain kinds of stress, including the kind of stress that might break DNA, will generate a stress response that causes specific variations in the genome. Now, she saw actually that genes could jump, from one part of the genome to the other under stress, which people found incredible. But eventually, the mechanism of that was worked out, that enzymes were induced that could move pieces of DNA around, transposases. Evelyn Wittgen also found a kind of global stress response in bacteria which Radman called the SOS response, meaning because his father had been a sea captain, save our ship.
Okay.
that when the organism was, for example, irradiated with ultraviolet light and there was a lot of DNA damage, there was a stress response that would, for example, copy DNA. It would get across some of these damaged places and it would not do it as accurately because the accurate copying mechanism would just be blocked at those damaged spots. And so it would be inaccurate copying. It would generate variation in the descendants. And it was kind of a global response that did other things in the bacteria too. So, and there's increasing studies on the effect of that. So the bacteria senses, it has to sense that it's under stress, whether that's
right.
its DNA is damaged or it's under nutrient stress. It has to sense that. And then there's a downstream signaling pathway that will then affect a variety of systems in the bacteria, including the DNA replication, the accuracy of the DNA replication. Yeah.
Well, from what you've seen, is it a really complicated path to start from stress to adaptation, start to finish? Do we have all the steps in any kind of adaptation, or we just kind of we know generically, but not exactly?
Well, it's just slowly, it's research over time, so we're learning more and more. It's easier now that we have whole genomes because we can begin to, you don't have to just look, is this gene doing something that you can begin to look at the kind of wiring of what genes affect which other genes. We can look more globally at the process. So as with much of biology now, having the ability to look at the whole genome, we can sort out these signaling pathways, mechanisms, inputs, cascades of signaling, and then outputs much more efficiently. So we're learning a lot about that right now.
What about the, make it even more complicated, but what about our microbiome? Perhaps that, it seems like bacteria are able to change their DNA and, morph much more quickly than eukaryotic cells, or somatic cells or germ cells. I don't know. I mean, have people even looked at how the microbiome could be playing a part in our response or any creature's response to stress and helping to assist the adaptation?
Yes, I mean, it's very interesting. We knew for years, I think every biology student, and even if you didn't take biology, knew that termites didn't really chew the wood. They had to have bacteria inside them to digest the wood, and that they were dependent on their bacteria. And so we knew that for termites, but we never realized that we also, had a lot of bacteria inside us, and that those bacteria were doing things. And now we know, and there's even been discussion of bacteria. First of all, they interact with each other. So one bacterium can make a compound that the neighbor, it might diffuse out and the neighbor could use it and then make something else and then another, so that they share. They don't all, they're not all required to duplicate all of the intermediary metabolism and all the steps. So they're sharing compounds, they're interacting, and then what in the last, I would say, 5 years has become apparent is they also that we have neurons in our gut, and they can send out small molecules that can have effects on our own cells, signal our own cells. So that's a whole area of research you might want to find. I could recommend later. Some people might want to talk about that on the podcast, but how the microbiome interacts with our cells and our even some neurons. So with our metabolism yeah.
Yeah, I was going to ask you this way early on, but knowing what you know, how does your brain not melt down when you think about the complexity of of living creatures, humans, otherwise? I mean, there's so many factors, so many things going on. It's just like unbelievable.
Right. So one really important concept is organization and hierarchies and of organization and regulation. The genome is definitely organized into different regions. Different regions are copied at different times. They're exposed to different probabilities of different kinds of variation. It's an area that we're just beginning to understand. but it's not like genes are randomly strung along in the genome. There are regions. There's organization, and there's levels of organization. There's hierarchies of organization. Another important concept, actually, we introduced with John Doyle, who's at Caltech, is it's the concept in engineering. Protocol is a term used in engineering for a set of rules by which components interact to create new levels of function. So there are protocols. I mean, a simple one would be the table of codons, the genetic code. So any gene that anything, we can get a gene from outside. A genetic engineer can put a gene in, as long as it follows the protocol of the table of codons, it will be expressed in the cell. There are more levels of protocol and regulation in a kind of hierarchical organization, and that's the only way everything can possibly work. And we don't understand. I mean, there's so much we don't understand yet about these many levels of organization. Or, I mean, you have to think about the whole idea of a single fertilized egg dividing and then dividing again and then dividing again and eventually becoming a person. That is, when you think about it, it's very, it's awe-inspiring that can happen. And I know I used to, I mentioned the American Museum of Natural History. I used to they have a slice through a sequoia tree there, which is, one of the mountain sequoias. They're huge, people would dance on them in big groups. They're very big. But they also had right in front of it, they had a sequoia seed, which is just a tiny little thing, maybe a quarter inch or something. And so you have to think about organization. I mean, what was in that little seed that could give you that big tree?
Right. Yeah.
Yeah. So the concept of organization you bring up, how can all this happen?
I know, it's crazy.
It's just there's a lot that we need to appreciate about it. We take things for granted, but there's a lot to learn.
Well, I'll ask you one last question. This is a real softball one. Okay. So, from a biochemistry perspective, how did life begin?
Yes. So there's a variety of theories. I mean, when I was in high school, there was the famous experiment where they took the primordial soup as they thought it existed then and passed lightning through it. Julia Childs did a beautiful version of this at the Air and Space Museum. I don't know if they still have that video, making a primordial soup. But since then, yes. And because what happened is they took the compound, what they thought was present, and then they irradiated, and they found compounds formed that are, they found amino acids and some nucleotides. They found things that are, quote, the building blocks of life. Now, life didn't crawl out of the experiment, but they said, look, you can make these molecules this way. But now there's all kinds of other theories, including that life might have begun down at the vents in the ocean, deep in the ocean, that where there's a lot of sulfur as a source of energy because there's not a lot of sunlight down there. There's many different theories about how you get from this inorganic molecules to life forms, but there are many circumstances now where people have seen that these molecules can form, and then there's a matter of structuring it and organizing it, whether it's on a clay, framework, or how you isolate and create a cell. But that's certainly not a settled issue. But for those who are interested in that, a lot of the astrobiology groups are interested in that too, because how would you define and recognize life in a different setting?
That's true. Yeah, that's true. Boy, so many questions, but this has been a great call. I really appreciate all your knowledge. So, how can listeners that are curious about, some or all of this stuff, how can they keep going and find out more? How do they reach out to, find out more about your work or reach out or, continue their education?
Well, one thing, I wrote Darwin in the Genome for a general audience. I've been told by some of my friends it's not a beach read, but they have read it, and I've tried to design it so that you can skip over some of the more complicated parts and pick it up, and it's not, if something gets frustrating, just try to continue a little later. So Darwin in the Genome is a good place to start. For those who are already scientists, computational biologists, biochemists, I also edited a volume, The Implicit Genome, and that brings up this issue of hidden and new forms of information that are in genomes that are not as obvious as they might be. And then, they're not just these linear sequences. And then finally, this issue of the feedback loop itself. There is a technical article I've written with John Doyle entitled "In Darwinian Evolution, Feedback from Natural Selection Leads to Biased Mutations." So those are three different levels of depending on how curious people are. Yeah.
Yeah, that's great. That's great. Well, excellent. Well, Lynn, thank you so much for coming. This has been an awesome call.
Well, thank you. I appreciate your curiosity and enthusiasm for these ideas.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.